Hello everyone, my name's Catherine. I'm going to tell you a bit about my work today in the Chagos archipelago, alongside that of my colleague and collaborator, Professor Rob Dunbar from the University of Stanford. So our work is focused on the importance of the coral reefs in Chagos, and we think that the reefs are important for three main reasons, and that is their biodiversity, their connectivity, and their productivity. And so that's what our work is broadly split into. So we have four main objectives. Our first is to look at the biodiversity of the understudied hidden fauna within Chagos reefs, and also what environmental variables might be driving this biodiversity. Secondly, we're looking at how connected the coral reefs are in Chagos, and also looking at this idea that Chagos might be a stepping stone across the wider Indian Ocean. And thirdly, we're looking at the reefs as hotspots of productivity, looking where these hotspots may be, and also seeing whether there might be potential export to other systems. And finally, how important are Chagos reefs to the pelagic biodiversity? And today I'll be focusing on the first three questions, as these are what we've managed to get underway in the first 8 months of the project. So to start with, looking at the biodiversity of reefs on Chagos. So what we term the cryptofauna, which is the small invertebrates and vertebrates which live within the reef structure itself. And they've been quite overlooked in Chagos, really. They make up some of the largest of the biodiversity there. And we know from some of our previous work that actually the decapod diversity on these dead coral heads, which you see up here, is some of the highest recorded globally to date. And also about 35% of the cryptofauna are rare species. And by rare species, I mean only one individual was found. And these guys here, these four species, were the most dominant that we found on these coral heads. So 2 species of Hippolytidae shrimps and 2 species of squat lobsters. So we're continuing this work now using more standardized structures. So obviously coral colonies have intrinsic variation, and that makes it hard to get standardized measurements of the biodiversity. So instead what we'll be using now is these autonomous reef monitoring structures, which you see here. So we've put these out at 4 locations across the archipelago on the expedition in March this year. And the idea is that these get left out on the reefs for a period of 1 to 3 years. And over that time, they become colonized by the small motile and sessile organisms which live within the reef. And then we collect these and we process what's colonized the structures. So in addition to this, we're also looking at the plankton diversity on these reefs. And as you will know, these comprise the base and the lower levels of the marine food chain. And in turn, that supports everything else, including the commercial fish species. They are also important food sources for heterotrophic corals. And their diversity has never been assessed in Chagos waters. So to assess the biodiversity of the plankton, we're using environmental DNA. And I noticed there was a question in the session before asking if anyone was looking at this, and we are, is the answer to that. It's early days, but to date we have taken 32 samples from around the archipelago, highlighted by these green dots here. And we do that using non-invasive water sampling, using these Niskin bottles which we drop over the side of the boat to collect our water samples. These samples are then taken back and we process them using metabarcoding techniques, and that will allow us to see the diversity of the plankton organisms within the water column and look at the spatial and the temporal diversity, and also we'll be looking to see what will be driving these biodiversity patterns. So to do this, Rob has put out oceanographic instruments across the archipelago, many of them at the same sites as which we've been doing the biodiversity monitoring. And we started this work in 2016, and last year we were able to collect the majority of these instruments and also to replace them and add to the sites where we've had these out previously. And you'll note here that in the green, many of those match up with the sites where the autonomous reef monitoring structures are. And we'll go back next year to collect those instruments and download the data and then hopefully replace those on the reefs. So this is just a snapshot of some of the data which Rob has been processing from these instruments. So this is from the temperature loggers that we've had out. And this data is from March 2016 to February 2017. And this really just illustrates the seasonal variability in temperature across 7 sites at the archipelago. So we do have data from 20 sites, but to date, this is just showing you 7 of those sites. And then if we zoom in on this data set, this is just looking at 5 days worth of the temperature logger data. And this really illustrates these large plunges in temperature which you're seeing at some of the sites in Chagos. So not all of them, some of them are quite constant, as you will see up there, but all of the sites at Salomon and one site at Perispanos has shown these large plunges down to about 21.5 degrees. And this is something that Charles Shepherd has shown previously with his temperature work in Chagos. I think what we're adding to this here is that this data set has very high resolution. So this data is taken every 15 seconds, and that's hopefully going to allow us to diagnose what is causing some of these temperature plunges at some of these locations. And I think Phil mentioned in the previous session that as biologists, we don't talk enough about internal waves. Well, we think that it is potentially internal waves which might be causing these temperature plunges. He'll be pleased to hear. So this is important because it probably shows that many of the coral reef organisms in Chagos are having to adapt to their local thermal environment. And so we think that there's probably site-specific thermal adaptation which may infer some resilience to heat stress in some locations. And that makes, I think, the question of the connectivity of some of the coral propagates even more important between the different locations within Chagos. And that leads us on to this next question on looking at the connectivity of Chagos's coral populations. So we're looking at this within the Chagos archipelago, and that we'll then be extending this out to further into the wider Indian Ocean. So we know that the Indian Ocean harbours about 13% of the world's coral reefs, and that about 65 million people live within 35 kilometres of the coastline. So this makes these reefs extremely important for people's livelihoods. And Chagos is hypothesized to be very importantly placed within the Indian Ocean, obviously smack bang in the middle geographically, and also because of the prevailing currents moving west to east and changing halfway through the year. So potentially Chagos corals could be very key coral populations for maintaining the health of reefs in the Indian Ocean and also for reseeding them after damaging events such as the recent coral bleaching events we've seen in 2015 and 2016. So on this year's expedition, we collected some samples of 3 coral species across the archipelago in Chagos. And these 3 species were Acropora cytherea, which are these very characteristic table corals, which you see here. Porites lutea, the massive corals here, and then Acropora tenuis as well. So these species were mainly chosen because they're some of the major reef builders in Chagos. So we've collected samples of these corals from 20 sites across the archipelago, which you'll see with these red and blue dots on the map. And we've collected very small samples in order to reduce any ecological damage to the reef as well. We now have these samples back in the lab in Oxford, and we're starting to undertake the genomics work, which we need to do in order to investigate the population structure. So at the moment, we are isolating the DNA, and we're also quality checking the DNA before we move on to these next steps. And this image here is an example of one of our gel electrophoresis images. So this is how we check the quality of the DNA. And what we're looking for is these very high, tight bands that you see here. And that indicates high molecular weight DNA. We're not so interested in these sort of smears of DNA, which are lower quality. So generally, about 3/4 of these samples have good quality DNA for us to continue using, which is pretty good for corals. They're quite tricky, non-model genomics organisms. So in addition to this, we're working to extend this work across the Indian Ocean. With colleagues and collaborators, we have samples from the Seychelles, also from the east coast of Africa, and we'll be looking to get samples from other locations in the Indian Ocean, particularly some areas to the east, Chagos, Kili Islands, and the Maldives as well. So that's a work in progress at the moment. On to our third question. And that was, what are the hotspots of productivity and potential export to other systems throughout the archipelago? We do this using what are called benthic ecosystem and acidification measurement systems, otherwise known as beams, which you see here. And these allow us to directly measure reef metabolism and calcification using physics and chemistry. So this video here is showing you a deployment at El Anglaise in Salomon, which we did on this year's expedition. So these were deployed quite shallow this year, at about 2 meters, in order for us to pilot these systems out in Chagos. And this is Rob's team just deploying them here. And you can see this is in an area of reef which still has some really nice aquapora colonies, even post-bleaching from 2016. So these systems are made up of a few different instruments. They're made up of pH meters, of temperature, salinity, and oxygen meters, of light meters and current meter. And then there's the pump, which pumps the water across all of the instruments. So this pump pumps water from 20 centimeters for 15 minutes, and then it alternates and pumps water from 70 centimeters for another 15 minutes. And it does this continuously, in this case for a period of 48 hours. They were left out. So this is just some preliminary data from the beams, which we're really pleased with. They've worked well. And you can see here the light measurements, which is what you'd expect, this increase in photons during the day, and the temperature measurement as well, as you can see, a lot more stable than at many of the sites in Chagos, which I showed earlier. And then some data on the pH and O2, which we'll look at in more detail here. So this graph shows as the light starts to reduce, as we reach dusk on the reefs, the oxygen is starting to decrease, as you would expect, as photosynthesis decreases, as light decreases, and therefore less oxygen is produced. And with that, a function of pH also decreases. And these shaded areas here show when the pump is pumping water from 20 centimeters just off the bottom of the reef. And again, at that point, you have these peaks in O2, which is what we'd expect. That's where the majority of the photosynthesis is taking place. And then the light areas show the 70 centimeters above the water column pumping. And then if we zoom in again, this is just to illustrate that these gradients in O2 and pH are actually quite high. And here we're looking at the current data. So this is from the current meter that was out with this beam system. And this is, again, showing us exactly what we would expect, that the the current speed is increasing with height from the substrate. So all this information is used in this equation to work out the net community calcification and the net community productivity. And really what these figures are illustrating is that there is a net production of carbonate still in the system, and also a net production of food, which can potentially be exported as well. So this is good news for the reef, and this is just a pilot study at the moment, which needs to be scaled up. These need to be left out for a longer period of time so that we can reduce some of the daily variability and get more reliable measurements. But we're really pleased with how well those have worked. So just to sum up, our next steps will be continuing with the coral connectivity work to analyse our samples and also extend this work out to the wider Indian Ocean. We'll be examining some of the biodiversity data which we've collected and be able to identify the environmental variables which may be driving these communities, deploying the arms for a further length of time in the archipelago, and potentially merging this data with other estimates of reef growth, and then also addressing our fourth objective to look at the offshore food exchange and if these shallow systems are potentially very important to the pelagic biomass as well. So thank you all for listening. Thank you to our collaborators and to all those who support the expeditions, and of course to the Bertarelli Foundation for funding our work. Thank you.